Hey guys, welcome back. This is Lei. In the last video, I showed you how to analyze small RNA sequencing data. Small RNAs are composed of many RNA species. Among them, microRNAs are probably the best studied small RNA species. They are derived from their hairpin precursors and then form the RNA-induced silencing complex RISC to regulate gene expression or translation. MicroRNAs are involved in many biological processes or diseases, and many of them are used as drug targets. In this video, I'm going to show you how to analyze microRNA sequencing data in two ways. As usual, our first step is to set up our conda environment. For people who are new to my channel, conda environments are Linux environment. So you just need to open your terminal on your Mac or Linux computer and copy the code in the slide. So the environment we'll use is the RNA sequencing environment we created before. And we're going to need a new tool called miRDeep2 specifically for microRNA sequencing data. The aligner we're going to rely on is still Bowtie. As I said, Bowtie is old, but it's very good at handling short reads. So just make a new folder for the Bowtie index. Because microRNAs are so well studied, they have their own references. So you just need to download the microRNA references from miRBase using wget command and URL. The mature.fa is a microRNA sequences reference. The hairpin.fa is the precursor reference. And of course, we're going to need human reference genome. Our data is human data, so if you have other species, just download the corresponding references. So this is the format of the genome or reference file. So it starts with the greater than sign and the name, and then the second line is the sequence. So this is a mature.far. It's the microRNA sequencing reference. So the file we downloaded contains the microRNA sequences for all the species, but we only need human. So our first step is to subset or extract human sequences from the mature or the hairpin .far. So we use awk, another language or command, to subset the microRNA reference. And the next, we removed spaces from the title of each sequence using the remove white space in id.pl. This command is from miRDeep2 we just installed. So we just do the same thing for all three references and save the result to a new file called mature HSA renamed.far. And this greater than sign is the output sign. So you output the result to a new file for all three references. And here is the catch. Notice that there are many U's in the sequences. They are actually, because they are RNAs, they should be T's, because you're going to map the reads to these sequences. So we have to replace all the U's with T's. If you don't do it, you'll get the mapping rate 0. So I learned it hard way, so don't make a same mistake. So we use set command to replace all the U's with T's in all the files here. And then we build our Bowtie index the same way we did it in the previous tutorial, using Bowtie build and the reference genome, and then the directory of the index. And then we build another one for human genome in the same way. This one for the microRNA reference. So once everything is set, our next step is to trim the adapters, just as the same thing we did for a small RNA using trim galore. And the adapter sequence is from the sequencing kit you use. Just make sure you have the right adapter sequence. And then we save the result to our directory we create for each sample. And here is the FASTQ file we have for our data. Remember, microRNAs are very short and small. It's like 18 to 22 nucleotides. So don't do pair-end sequencing. You're wasting your money. So the length of one read is like 75 to 100 nucleotides. So you don't need to do pair-end sequencing. And once we have the cleaned or trimmed FASTQ file, we can do the alignment using Bowtie. So the settings are exactly the same as we did for the small RNA sequencing. So these options are for the best mapping. The -X here, we use the microRNA reference as our reference here. -P, we need 16 cores. -Q to specify our trimmed FASTQ file. -S is output. The output for Bowtie is SAM file. So save it as SAM. So next, we convert SAM to BAM using samtools. So first, convert SAM to BAM like this. And then we sort the BAM file and save to a new file. And then we index the BAM file. These steps are necessary for quantification. So next, we do samtools flexset. Remember in our last tutorial, this command is to generate mapping rates, the quality control file. And this is the content of the file. So let's check the second line. The alignment rate is 82%. It's perfect. And next, we can directly quantify the microRNAs using samtools, the IDX-stat option. So we can save the result to our output file here. So you can also use feature counts like we did for the regular RNA sequencing to quantify microRNA expression. But you have to generate or create a customized microRNA annotation file, the GTF file, as we did in the small RNA sequencing tutorial. But things are getting complicated, right? Because you have to do so many things to quantify it. As I said before, as a programmer, we want to be as lazy as possible. If things get too complicated, maybe there's another way, a simple way to do it. So next is a simple way. The method 2. We're going to use miRDeep2 to quantify microRNAs in 2 steps. So first, we call the mapper.pl to map our FASTQ files directly to the human genome. Here, we can use our the trimmed FASTQ files. And here, -G, that means the genome. We use the human genome, HSE. -L, the minimum length, 18, because the microRNAs have like length from 18 to 32. And these options, you just keep them as they are because they are the options to specify the mapping accurately. The -K is adapter, so you don't do the trimming if you specify adapter here. So you can just use the original, the raw FASTQ file over here. Specify the adapter sequence. And -S is the output, the collapsed FASTQ. This file has a collapsed reads. That collapses all the reads, the identical reads into one ID. For the next step, -P to specify the Bowtie human genome. -T is the alignment file. It's comparable to the BAM file, the ARF file. So we run the first mapper, and then we use the output from the first step to quantify the microRNA expression using miRDeep2.pl. So the first line, the input is the collapsed FASTQ from the first step. So .fa is the genome, the human genome file we specified here. Next, we specify the alignment input from the above step, the ARF file. Next, we specify the microRNA reference file, the .var. And the second one, I specify none. That means, well, you can specify a closely related species like chimpanzee, microRNA reference. I don't have it, so I just don't want to do it. You can specify, leave it as none. Next, specify the precursor. So we have this hairpin precursor reference. Next, -t HSA, that means human. So you have mouse, you can use other symbols over here. So these are the results in the folder, many files. But we only need to pay attention to the expression file. So just find them and open it. It's TSV file. You can open it with Microsoft Excel. And this is a comparison of the two methods for microRNA quantification, samtools and miRDeep2. As you can see, the results are very similar. Maybe some of them are a little bit different, but they're quite similar. So it's your choice to use any of the methods you prefer, but why not using this one? Just 2 steps, right? Okay, so that's the whole tutorial for microRNA quantification. The details are in my website, the corresponding post. You can copy the code in the post directly and try them. And don't forget to check out my other website, bullishbooks.com, for personal development. And subscribe if you like it. And thank you for watching.